And I'm back. Wi-Fi kicked me off. I had to go down, acquire an Ethernet cable, and now we should be okay. I shouldn't be kicked off the wireless anymore since I'm now hardwired into the building. So let's pick back up with temperature stability. Again, as I said before, I was so rudely interrupted. Temperature stability is water's ability to maintain temperature. It takes an awful lot of energy to remove or to raise the temperature of water. There's a picture. The top picture is a picture of a water molecule, a single water molecule. And as you can see, I don't know how well you can see it on this. Let me try to dim these lights in the front of the room just a little. Maybe that will help. Yeah, looks like it does. Sorry about that from the front half of the meeting. Okay, I'm about to trip over my new cable. There we go. Lovely. You see your instructor fall on the ground. Nobody laughs. All right, but you see here you got a slightly positive end to the molecule, a slightly negative end to the molecule. And the slightly negative end of the oxygen and the slightly positive end of the hydrogen are sort of pulled together in an attraction. That's where we get our hydrogen bond. And then here's a whole bunch of water molecules held together by hydrogen bonds, but you can see there's all these hydrogens and oxygens that are left unattached and available to form even more bonds. And that's one of the things that makes water very beneficial, gives it some of its really unique characteristics. I also used the time looking for an Ethernet cable to get a couple of more markers, so maybe we'll be a little better off now. Properties of water, again, water molecules form hydrogen bonds. They repel nonpolar things. Things that are water-loving, we refer to as hydrophilic. These are substances that when placed in water will dissolve very readily, like sugar and salt and those kinds of things. Hydrophobic things are water-dreading. These are substances that tend to be nonpolar. Oils, as anybody knows, oil and water don't mix. If you happen to like a salad dressing that has vinegar and water in it, it's always vinegar and oil. It's always kind of layered, and you have to shake it up really good to get everything mixed together. And then you pour it on your salad. You set the bottle down, and the next time you pick it up, they've separated again. That's because the oils are hydrophobic. Water, again, cohesion, the tendency of molecules to stick together. Water evaporates. When it does that, it absorbs heat. That's called the cooling effect. Again, it's how our body cools itself. You sweat, and as the water evaporates off your skin, it draws heat with it. Excellent solvent properties. Again, salt, sugars, and many polar molecules dissolve. Anything, when you have a solution, you have a solvent and a group of solutes. Kool-Aid. Kool-Aid, the solvent is water. The solutes are the stuff that's in that envelope. You tear the envelope open, you drop it down in there, you mix it around, and you get what is called a solution. A solution is a homogeneous mixture. It just simply means that everything is evenly dispersed throughout the solvent. Kool-Aid, for instance, you put the old-fashioned kind of Kool-Aid, you put the envelope and a couple of cups of sugar. There's new types of Kool-Aid now where everything's in the envelope and you just dump it and mix it up. But the solutes would be the sweeteners, the flavorings, the colorings, all of those things that give Kool-Aid its wonderful descriptive attitude. Grape Kool-Aid is purple. I had a little brother that wanted red Kool-Aid. He didn't care whether it was cherry, strawberry, it just had to be red. He wanted the red one. He first started talking when he was 2 or a little bit older than 2. My adopted daughter, Lindy, drinks green apple, and she calls it bug juice because I made the mistake one day of calling it bug juice, and she thought that was incredibly funny. So now whenever she wants something to drink, she just hollers bug juice, bug juice, bug juice. And we give her some green apple Kool-Aid. The reason it's not Kool-Aid, it's Jolly Rancher is the one she drinks. But again, the solutes are the sweeteners, the flavorings, the colorings, and the solvent is water, and then the solution itself is the whole entire drink. Temperature stability, again, water absorbs a lot of heat. Hydrogen bonds are what holds ice together. Temperature is actually a measure of molecular movement. The warmer something is, the more movement is involved in the molecules. The molecules are vibrating, and the hotter you get it, the more they vibrate. And if you heat water enough, you vibrate it to where molecules will start to fly off, and that's where you get steam. There's the ice, the lattice work of ice. You see the nice symmetrical way all the bonds are forming because we've slowed down the movement so much. This picture down here, again, if I'm looking at my camera, I'm looking at my computer screen, and it actually looks a little better than it looks to my eyes right here. But these are two goldfish. There's a couple of goldfish underneath the ice in somebody's fish pond. So you've got this nice lattice work of ice on top. Below the ice, there actually is still liquid water. Water separates into hydrogen and hydroxide ions, so you've got H and OH. It's a measure of pH scale is how we measure this. That is the measure of the concentration of hydrogen ions in the solution. The more hydrogen ions, the lower the pH. So if you see something with a pH of 2 and something with a pH of 3, the thing with a pH of 2 has more hydrogen ions flying around than the pH of 3. Pure water has a neutral pH, which is 7. Pure water is exceedingly rare outside of a lab. Seawater is not pure. Lake water is not pure. There are some spring waters in naturally occurring springs that is pretty close to being pure, although human beings have managed to mess some of those up to where that water is not pure anymore either. But basically, when you have a pure water, the number of hydrogen ions and the number of hydroxide ions is equal. An acid is a substance that releases hydrogen ions. It has a pH of less than 7. A base, another word you can hear for base is an alkali or alkaline substance. These are substances that release OH ions and pH of greater than 7. So things lower than 7 are acids, things above 7 are bases. This is a wonderful little chart that shows the different items. You have battery acid down here at the bottom, you have drain cleaner at the top, and in the middle you have all sorts of readily recognized things. Urine, for instance, has a pH of between 5 and 6. Pure water is 7. Milk is actually pretty close to 7. It's a almost neutral substance. It's one of the closest things that occurs in nature other than pure water to be neutral. You see, seawater is a basic substance. Most rain that falls, it has an acidic pH. It's referred to as acid rain. Some places it's worse than others. But there's all kinds of different things that are on this chart. Soft drinks are way down here at about 2, but your gastric juices, the juices in your stomach, are down around 1. So again, all kinds of different things. You can look on there and find your favorite item to see whether it's actually an acid or a base. Buffering system. Most molecules of life have a way to control pH. They have things called buffers, and if the pH gets too high, the cell will release a buffer to bring the pH down. If the pH gets too low, there's another buffer that will bring the pH up. So those are buffers. They're chemicals that keep the pH stable. Here's just an example that the bicarbonate acid in water, CO2 forms carbonic acid when you add it to water. The bicarbonate buffering system. Don't worry, I'll never ask you to complete these different chemical equations. Just know what a buffering system is. It's a way to control pH. Organic molecules. Organic molecules are things that are made up of carbon, hydrogen, and oxygen. Organic molecules are so important that there's actually a whole entire field of chemistry called organic chemistry. But these are the molecules of life: carbohydrates, proteins, lipids, and nucleic acids. They are all organic molecules. Something organic is a type of molecule that consists primarily of carbon and hydrogen and then you're also going to have some oxygen thrown in there as well. But when you just talk about sheer numbers, most of organic molecules are carbon and hydrogen. Carbon atoms bond covalently with up to 4 other atoms, often forming long chains or rings, and I'll show you some rings and chains here in just a minute. But the important thing to remember about carbon is it always forms 4 bonds. There's always going to be 4 bonds with carbon. Other elements may form 3, may form 4, may form 5, just depending on what they're combining with. Carbon always forms 4. There is a nice little, this is a chemistry shorthand way of drawing a molecule. This is telling you that there's carbon, 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 carbon there. What it doesn't show you is, yes, there are also a couple of other molecules hanging off there, or a couple of other atoms. It's not showing those in that particular shorthand. Everybody recognize this. It's a donut. Donut is made primarily of carbohydrates. And this is a wonderful way to get carbohydrates in your diet. Probably not the best carbohydrates that are possible in your diet, but they're carbohydrates nonetheless. Cells assemble large polymers from smaller monomers and break apart polymers into component monomers. A monomer is a single structure. And we'll just draw it like that, and only now I'm simply going to erase that because I want to actually turn it 90 degrees. I want it to be positioned that way. Okay, that is a monomer. That is a single unit. Even though it is made up of, a lot of different atoms, it's still a single unit. So we call it a monomer. These can be simple sugars like glucose and fructose and maltose and all those other oses. They can also be fatty acids, amino acids, nucleotides, all different kinds of things, a monomer. Just because it's a monomer doesn't necessarily mean it's small, just that it is a single unit. A polymer, on the other hand, is a group of monomers. And I'm just going to draw about 4 of these little monomers together to make a polymer. So each one of these individually is known as a monomer. Collectively, they are called polymers. Now, enzymes. Enzymes are products that speed reactions or help reactions along. An enzyme-driven reaction is one where for the reaction to occur consistently, enzymes have to be present. Now, it could be that there's an enzyme that's going to break each one of these bonds holding these monomers together, so we have an individual monomer. Or there's another enzyme that's going to attach this one to this one and form this bond. Those are enzymatically controlled reactions. And the sum total of all the enzymatically controlled reactions that take place in your body or in the body of a living thing all day, every day, is referred to as your metabolism. The way you break down food and reassemble it into fat or muscle or some other substance, the way your body controls blood pressure and temperature and heart rate and all of those things all play a role in your metabolism. The carbohydrate is used as an energy source for structural materials. You have to take in carbohydrates and then you reassemble those products produced from the carbohydrates to make others. You're going to make, you and I make primarily glycogen, but there are some other things we make as well. The simplest of the carbohydrates are the monosaccharides and disaccharide sugars. Again, glucose, fructose, lactose, sucrose, dextrose, maltose, all of those are monosaccharide and disaccharide sugars. Complex carbohydrates, by contrast, are combinations of simple sugars. You take all those simple sugars and put them together into big long chains, and you make a complex carbohydrate. Glucose monomers, for instance, can bond in different patterns to form different complex carbohydrates. There are actually 4 major complex carbohydrates, one of which is not a nutritional substance for human beings. It's found in our diet in a few places, but it's not a product we can use. We can't process it. If you consume some of this 4th one, which is called chitin, C-H-I-T-I-N, if you consume it, it just goes straight through your system. You take it in here, you get rid of it, the other end of the digestive system, and you don't process it. So we're not going to talk anything about it other than to just mention that it exists. However, the 3 that are parts of our diet are cellulose, starch, and glycogen. Cellulose is the structural component of plants. The major part of the leaves, the major parts of stems. If you eat celery, for instance, celery is almost all cellulose. Celery is one of the few food items that human beings can consume that has a negative net calorie. What that means is, if you eat celery, it takes more calories for your body to process it than it does for you to when you consume it. There used to be a diet, I don't know whether it's still one of the diets you can see on the internet when it says lose 50 pounds quick. But there was a thing where you made celery soup. You just got a whole bunch of celery, you chopped it up, you put it in a big pot with water and some salt and maybe a little pepper, and then you made that up into a soup, and then you could just eat as much of that as you wanted to. You ate 24 bowls of it a day, didn't matter. As long as your body would hold it, it would, because you were going to lose weight because it takes more to process it. My wife and I tried this diet one time many years ago, and it worked for me for about a week. And then after 5 bowls of soup a day for a week, I decided, well, I need to put some crackers with the soup. And then so I ate some crackers, and the amount of soup went down. And then after a few days of crackers, I decided a sandwich would be better. So, I made a sandwich to go along with my celery soup. And eventually, I went back to eating just exactly the way I was before, and the celery soup all went away. However, I have lost a lot of weight. I've lost over 120 pounds. Yay me. But it wasn't eating celery soup. But celery is almost all cellulose. That's why there's no calories in it. Cellulose is something we need in our diet. It's just something we don't process. Excuse me. We don't process it very well. Excuse me. Starch is the main energy reserve for plants. Potatoes, a lot of starch in potatoes, all different kinds of potatoes. There's starch in radishes and turnips and all of those root vegetables that we eat. Nature, again, did not make the potato for you and me to have French fries and mashed potatoes. Nature made the potato for the plant to have an energy reserve. When the rain stopped falling and the sun stopped going down and the plant needed some other way to have energy, it used the potato. That's the function of the potato botanically. And then glycogen is the energy reserve in animals. Up until just about 40 years ago, glycogen was referred to as animal starch, because animals use glycogen the same way plants use starch. We store it primarily in our livers, and it's there for energy when we stop eating. So one of the first things we start doing when you go on one of those crash diets is you don't start consuming fat. You start consuming all your glycogen reserves. There's a nice little picture on the upper left. You see the nice symmetrical form of that upper left drawing there. That's what cellulose looks like. Again, a very symmetrical, but each one of the little orange hexagons represents a glucose molecule. Then we have starch, and you see the way the starch, the starch is actually a long chain of glucose molecules. And what happens is, as the chain gets longer and longer and longer, it finally starts to coil. And you wind up with this long coiled structure in starch. And again, in glycogen, you see the way we make one long chain, but then along the long chain, we will grow these little side chains. So again, glycogen, starch, and glucose, all made up of the monomer known as glucose, or excuse me, that's cellulose, but all made up of glucose molecules just arranged in different ways. And this is a picture of a nice young man showing the cellulose from the palm tree, the trunk of the palm tree. I think that's a mango he's holding in his hand, showing the starch that's in that fruit, and then the glycogen that's supposed to be pointing toward his liver. Lipids are more commonly referred to as fats. These are those greasy, oily, waxy things. They often have one or more fatty acid tails. They'll have sort of a sugary-like head, and then the fatty acid tails running off the back. Lipids are, again, those things that are fatty, oily, and waxy. Fatty acid will contain a long chain of carbon atoms with an acid on the far end, or an acidic carboxyl ion on the far end of the chain, and there'll be a picture of those in just a second. Matter of fact, the very next picture is a picture of those. So at the top, you have the carboxyl group, or the head of the fatty acid. Then down toward the bottom, you see all of the different carbons and hydrogens. And if you'll remember, I told you that carbon always forms 4 bonds. And if you look at this first one, stearic acid, which is a saturated fat, if you count the number of items connected to each one of those carbons all the way up, you will see that each one contains 4. This one at the top is kind of unusual. You see that 2 lines there? That represents a double bond. That means carbon, that is called a carbon to carbon double, or excuse me, a carbon to oxygen double bond. But instead of 2 electrons being involved in that bond, there's actually 4 electrons involved in that particular bond. This omega-6 acid, linoleic acid, shows what is called a carbon to carbon double bond. And if you will notice, this acid right here, this fatty acid is nice and straight, very symmetrical. This one got these 2 bonds on it, shows that little bend in it. These acids, the saturated fats, are always going to be solid at room temperature. You have to heat them up to get them into a liquid state. Think about if you've ever cleaned a chicken and you got the fat hanging off the chicken, and if you put that fat that you peel off the chicken, if you slap it in a skillet, it'll melt. But you have to heat it to get it to melt. If you've ever cooked with lard, for instance, lard is rendered animal fat, and it is nothing more than completely saturated fats. When you start putting these double bonds in, you bend the molecule. When you bend the molecule, you make the molecules less likely to come together and form a solid. So most of these unsaturated fats are going to be liquid at room temperature. You got your omega-6s, you got your omega-3s, and you see, and then you've got this one that's just simply a cis transition, or cis conformation. What you notice, I hope you notice about this one, and this one, is at the area of the double bond where the 2 hydrogens appear over here and over here, the 2 hydrogens appear on the same side of the carbon. And when that happens in a chemical reaction, we refer to that as a cis conformation. Cis tells you that at the point of the double bond, the resulting hydrogens are on the same side. However, on the far side, the one you can't see very well because it runs off the edge of the too small screen, if you'll notice here, you can see this hydrogen on this side, but you can't see it very well, but this hydrogen's over here. They're on opposite sides. And in that conformation in chemistry, we refer to it as a trans conformation. This is a trans fat. This is one of those ones that human beings have manipulated. And these are the ones that we find out aren't so good for us anymore. And that's what happens when we make them. They don't show up looking like this. They show up looking like that. Fats are the most abundant source of energy in vertebrates. It is stored in adipose tissue. Adipose tissue, in addition to giving us a long-term storage of fuel reserve, it also gives us an insulator. Babies, for instance. Human beings actually have 2 types of fat. We have what is called brown fat, and we have white fat. Brown fat produces energy, produces heat. White fat is an energy storage. Babies little bitty babies, freshly born babies, have a high concentration of brown fat. So they can keep themselves warm better than we do. But as you grow up, your brown fat goes down and your white fat concentration goes up. The triglyceride is a lipid with 3 fatty acid tails. The triglyceride is the most common form of lipid found in the human diet. Again, there are other forms, but this is the most common. A phospholipid is a lipid that has a phosphate group attached to it, or multiple phosphate groups, depending on which one you're talking about. The lipid bilayer, the structure that is the foundational structure of all cell membranes, is a phospholipid. And phospholipid membranes form in a head-to-tail orientation. One end of this, the hydrophilic end, loves water. The hydrophobic end does not like water. So they arrange themselves with the heads up and the tails down, and the heads down and the tails up. You've got water outside the cell here. You've got water inside the cell here. So these two layers orient themselves, the hydrophilic end toward the water, the hydrophobic end away from water. A wax is a water-repellent liquid with long fatty acid tails bonded to a long chain of alcohol or carbon rings. Waxes naturally occur on the surface of lots of different leaves, for instance. And we all know bees make a wax for to make their hives. A steroid is a type of lipid with 4 carbon rings and no fatty acid tails. Steroids are very common molecules produced by the body. They have gotten over the last 25 or 30 years a somewhat undeserved bad reputation. Steroids are good. Steroid abuse, bad. Steroids, you have to have them. If you don't have steroids, you don't make things like estrogen and progesterone and testosterone and all of those things. Those are steroids. There's a difference. If you look, the structure of testosterone and estrogen are fairly similar. One of the most beautiful ducks that birds you'll ever see in Arkansas is the male wood duck. These are very pretty birds, and it is the presence of testosterone that actually causes this bird to develop the little tail he has on the back of his head, all the beautiful coloration. The female who produces estrogen is much more drab. It's actually a way nature ensures that the female, when she's sitting on her nest, will not be easily seen, because she's going to blend in. She's going to build her nest along the marsh line, and she's going to be in bushes and in grass and shrubberies, and so she's not going to be real visible while she's sitting in there. The male, while this coloration is very beneficial when it comes to attracting a mate, because the females pick the males that are the prettiest and have the prettiest courtship rituals, are the best courtship rituals, but this also makes him very visible to predators. He can't hide from predators very well. Protein is an organic compound, consists of one or more chains of amino acids. Its function is determined by its structure. Proteins are made. We're going to make muscle out of protein. We're going to do all sorts of things with protein. An amino acid is a small organic compound with a carboxyl group on one end and an amine group on the other end, attached to some other item. That R group could be any number of things, but the opposite ends of all amino acids look the same. They all have this NH2 on one end, and they have this COOH on the other end. And then the different amino acids, what this is what makes the difference. Amino acids are linked together in these long chains of proteins, and the type of bond is called a peptide bond. The peptide bond that forms amino acids, proteins used to be called polypeptide chains, and they still are in some fields of study. You and I refer to them as proteins. A chemist would refer to it as a polypeptide chain. Again, how protein structures arise. You basically have a a protein. This one on the far end is methionine. Again, here's the NH2. Actually, it's NH3 and the COOH. And here you've got this nice little differing group in the middle. Make the methionine. Then you make valine. You see valine looks different than that. Then you have methionine and valine linked together by a polypeptide bond, just right there. And then you add a histidine and a leucine and a thyroxine and a thymine. And I can't read what that last one is, but it doesn't matter. But you just add more and more and more amino acids, and you grow a bigger and bigger and bigger protein. Again, this is the second half. So as more and more begin to add, it starts to coil. The coil forms that chain of the chain of the green rectangles is referred to as a primary structure. Then when it starts to coil, you produce what is called a secondary structure. As the secondary structure coils and continues to coil and starts to form loops and helixes and all sorts of other things, you get a tertiary structure. And then you get these larger proteins where you see all the blue ones are one type, and there's some red ones that are a different type, and there's some green ones that are a different type. And all of those make what is referred to as a quaternary structure. And then you take these different proteins and you can make things like red hair, you can have large ears or big ears, all of those things that are the result of protein production. Changes in a protein structure may alter its function. We refer to that as being denatured. The protein has been denatured. One of the old instructors that used to work here many years ago was the chair of the department before I was. He always used to jokingly say he wanted the department to buy a flame-powered animal muscle denaturing device or something. Now, he had a big long name for it. What he wanted to buy, jokingly, was a barbecue grill. Because when you fry or when you grill, when you cook meat primarily is muscle tissue. Muscle tissue is made up primarily of proteins. And when you cook it, you're denaturing those proteins. You're changing the formation of the proteins. A prion, or as it was always pronounced prion. When I was going to school, and for years it was pronounced prion. The last 3 or 4 years, I've started hearing people pronounce it prion. So prion, whichever one it is, they are a misfolded protein that becomes infectious. The example of a prion disease that almost everybody has heard of is mad cow disease. Bovine spongiform encephalitis. Yeah, just remember mad cow disease. When I ask you for the most well-known prion disease, you're going to say mad cow disease. There is a human example, which is called the variant, which is a little v. Anytime you see little v in front of something, that means variant of Creutzfeldt-Jacobs disorder. Creutzfeldt-Jacobs disorder is a condition that occurs in the brain of human beings where your brain literally starts to, on a scan, looks like it's full of holes. Well, it's not actually full of holes, but it's just these prions are forming, or prions are forming, and they start to make big masses, which are referred to as plaques, and those plaques show up on a brain scan as a big dark area, so it looks like the brain has a hole in it. And the reason it is called the variant is there is a form of Creutzfeldt-Jacobs disease that you can actually inherit from your parents. It is a genetic disorder. But there is another one where through life a mutation occurs, and when that mutation occurs, you have what is referred to as the variant of Creutzfeldt-Jacobs disorder. Nucleic acids, nucleotides, the monomers these are the monomers of nucleic acids. Each nucleotide has a 5-carbon sugar, a nitrogen-containing base, and a phosphate group. The polymers and monomers are joined by a sugar-phosphate bond, and these function as energy carriers, as enzyme helpers, as chemical messengers, and as subunits of DNA and RNA. So lots of different things that nucleic acids can do. When we think of nucleic acids, we think about DNA and RNA because their names: deoxyribonucleic acid, RNA, ribonucleic acid. But there are other nucleic acids as well. And again, when we talk later on in the photosynthesis, and we talk about something called FAD and NAD, those are nucleic acids. But they serve as energy carriers at that point. There's a picture again of the nucleic acid. Here's the ribose sugar. Here is the phosphate group. Here's the base. Therefore, if that's ribose sugar, this must be DNA. Excuse me, it must be RNA, ribose sugar. But then you take this RNA and you take its complementary chain, turn it over that way, you make DNA. DNA consists of 2 chains of nucleotides twisted into a double helix. So, what is a prion disease? Prion diseases all affect the brain and the neurological system. They're difficult to detect and treat because it takes protein therapy, and protein therapy is in its very early stages. Cholesterol gets a bad name because too much can cause health problems, but you have to have cholesterol. Without cholesterol, you don't have a lot of the other steroids. Cholesterol is the cake mix of steroids that you have to have. The body takes cholesterol and adds and subtracts and makes other steroids. If you don't have cholesterol, then you have an issue. If your cholesterol is too low, one of the things that I discovered when I went on, when I found out I was diabetic, and I really changed my life. I changed the way I was eating. I changed lots of things. And one of the things they do when you start a diet, working with an endocrinologist, is they don't just check your blood sugar. They check lots of other things as well. And my cholesterol, which had always been kind of high in my adult life, my cholesterol through my change of life went so far down that the doctor said, you may not even be making, we need to check your testosterone because you may not be making testosterone. Your cholesterol level is so low. Well, luckily, my testosterone level was still where it should be, but I had to, because when I was taking a cholesterol medication, I was taking a blood pressure medication that also helped with cholesterol. I had to get rid of all of those and change everything. Of course, now, everybody pat me on my back again. I'm down to 2 prescription medications. At one time, I was taking, I can't hold my toes up, but I was taking 13 prescription medications. In my mid-40s, I was taking 13 prescription medications. I'm now 58 and take 2. And one of those is a diabetes medication. All right. Questions about that? If you have questions, contact me either in the message center or send me an email directly. Chapter 3 will appear in the next few days. And hopefully with Chapter 3, we won't have any technical glitches, which now I think I have everything I need to stay connected. So have a great day. See you next time.